Yes. Hello dear students. Let me start now a revision of genetics. First unit reproduction in organism, next sexual reproduction in angiosperm, and then the reproduction in human being, and as well as reproductive health also you studied. That topic already you studied in the class, particularly chapter number 1 and the chapter number 2 you studied in the class, and chapter 4, third also you studied in the class, and reproductive health, that just you can read out, and I think you did not require the revision of that. Genetics is very important, and that genetics I will start first. See, genetics. What is genetics? See, genetics means it is a study of gene. Study of gene. What is a gene? Gene is, as you know, that is a segment of DNA content. A segment of DNA is called as a gene. Segment of the DNA. So means in our body cell, the nucleus containing the DNA, and that in the DNA, the genes are present. Segment of DNA is called as a gene. And these genes particularly here help in transmission of character from parent to their offspring, and that is what you call as heredity. Called as what? Heredity. And this heredity is by what? Gene transmitted from one generation to the what? Another generation, one generation to the another generation, and this is what heredity. And from parent to their offspring, the expression is not exactly the same. The parent and the daughter or the son are not exact copy, and there is a little bit differentiation, and that is what you call as variation. And that is what? Heredity and variation. Variation result in evolution. Variation result in what? The evolution. So evolution means what? So evolution means change, decent change that taken place in the living being. Decent mode of change that taken place in the all living being, including human being also. Human evolution topic also you studied in the online class. So that is also one important chapter of your second year science. Now here heredity and the variation. So this heredity, what the topic, and this the main property, the first studied by Gregor John Mendel. First studied by Gregor John Mendel, and Gregor John Mendel, who is from 1956, he carried the several experiment on, particularly on his the pea plant, and that is Pisum sativum. Pea plant means it is a leguminous plant, just like beans, the peas, and then so many black gram, the green gram, what you see, and all such are called as what? The leguminaceae, family leguminaceae. They belong to the leguminaceae, and all leguminaceae plant, they bear legume as a fruit. Legume as a fruit. That is why they are called the leguminaceae. And such all the plant here particularly they produce a legume. And one such plant he selected, that is what? Pea plant, Pisum sativum. Why he selected pea plant only? That is already you studied. I am revising the taught the subject, because it can easily culture, quick result it gives, and it can carry self-fertilization without anybody's help, and it also having the contrasting character. Also having the contrasting character. So like this, so many important characters are there, and because of this reason, Gregor John Mendel, who selected only the pea plant for his hybridization experiment. So then, what is hybridization experiment? What is hybridization? So hybridization means it is a process of crossing of two parent, and the resulting individual is called a hybrid, and the process is called as what? Hybridization. But once again repeat now. Maybe it is asked in the one marks question, what is hybrid and what is hybridization? What is hybrid? When two parents are crossed, when the two parents are crossed, the resulting individual is what? Hybrid. And the process is called as what? Hybridization. The resulting individual is called as what? Hybrid. Resulting individual is called as a hybrid, and the process is called as a hybridization. So means Gregor John Mendel, who used the particularly for his hybridization experiment, two the contrasting character, just like a tall and the dark, the violet color and the purple color, so the violet color and the white color. So like this different character he used during his crossing, and so that process is only by the hybridization. So you understood about the hybridization. Now he selected pea plant, and in the pea plant hybridization experiment, he could get probably 36 contrasting character. 36 contrasting character. 36 contrasting character. Contrasting character means the 36 different pair of character. 36 different pair of character, but in his later experiment, he could use only 7 contrasting character. Only one? The 7 contrasting character, and such 7 contrasting character in which he later used, see the column it is given. See, and here the character. Character, if you like, the first character is a stem length or stem height. Stem height, and in which here one is dominant, and another one is recessive. That all it is given. Just go through your notes. Just I am just reminding you and revising. Here, dominant character means it is always one strong character. Dominant means strong. Recessive means it is a weak. Recessive means weak. Remember. And stem length or stem height. If I gave here capital T means it is a tall, and I can write symbol capital T. Correct. And recessive character is a dwarf and small t. So you like this is a tall, this is a tall, this plant is tall, another one is a dark. So like this, dark means small and the tall means bigger one. So means here the two character and T is called as a contrasting character. Contrasting character. Now the color of the flower. Color of flower. Flower color. So flower color is here dominant is violet, and that is capital V. Capital V. And then recessive is white, and that is small v. You can take small v. Next, color of the pod. What is pod? You know, beans, and it is a beans, and it is a beans, and this beans color generally is not green. No, dominant color is a green. So there is a color of pod. Pod means the green external surface, and that is generally is what green and capital Y. It is a capital Y, and then the yellow is it is a capital G, not capital Y. Capital G, green capital G, and for yellow small g. Dominant is taken on the small g. Next, here seed shape. Seed shape. See, there are two types of seeds are there. One is a complete round seed. This is a round seed and it is a dominant. And some other seeds are like this. They have to be very small and constricted. And see, seed shape is one is a round, and that can be written as a capital R, and another one is a wrinkle, and that to be small r. Okay. So then next one is a pod shape. Pod shape. See, this is a pod, this is a pod, and this is a correct shape of pod it is, and if it is so, and it is called inflated, and that can be written as capital I. That can be written as what? The capital I, and if it is it is not proper, and that is called as proper anthropoid. If it is the same is like this, if it is going like this, and that is called as constricted, small I. It is what? The constricted small I. Okay. So next one. So 5 characters is over, and the next one is position of the region, the main region is the what axial region. So axial is a axial, capital A. Axial is the main dominant character, and next one is terminal. Terminal is the small a. Terminal is what? The small a. So the sixth is the last one is that. The last one is a seed color. This is a seed color, and the seed color is generally yellow, and it's a capital Y, and sometimes there is also green, small one. So generally you see that what is a green gram or togari bada what you say, and that all color is generally is what a yellow, and the yellow is a dominant color, and the recessive color is what? The green. Recessive color is what? The green. So like this here the seven contrasting characters are there, and all these seven contrasting characters what is be taken from next hybridization experiment, and in the next hybridization experiment he want to cross with the one another, and then all the resulting plant he called as what? Hybrid. All the resulting plant he called as what? The hybrid. So in this way the hybrid plant is a resultant due to the crossing of the two parents. So this is a tall parent, dark parent, violet, white, green, yellow. So means these are the two parents, and the two parent if it is crossed, what we get? Hybrid. Similarly, violet and white also you cross, what we get? Hybrid. And that process is called hybridization. So with this the basic idea. Now let me go to the start. The first one gene inheritance. Directly I am going to the one gene inheritance. One gene inheritance. One gene inheritance, or it is also called as monohybrid cross. Remember this. Monohybrid cross. One gene inheritance or monohybrid cross. So then, what is this one gene inheritance? So one gene inheritance means here the single gene character that transmit from the parent to the offspring. Parent to the offspring. So monohybrid cross means also same. So monohybrid means crossing of two parent with respect to the only one contrasting character. Crossing of two parent with respect to the only one contrasting character, and such is called as monohybrid. Such is called as what? Monohybrid, and cross is called as monohybridization. Cross is called as what? Monohybridization. Understood? So one gene inheritance means what? Crossing of two parent with respect to the one contrasting character, and the resulting individual is called as monohybrid, and that cross is called as monohybrid cross, and the process is called as monohybridization. Now let us start. Now I take here only one character, or seven contrasting character already I have given to you. Seven contrasting character, in which any one character will start first. Let me start as it is given in the book, and that is a tall and means a dog. That is a length of the plant. The length of the plant we can take as a one character, but it is to be with the help of the two parents. No? So again, let us start. The first one is a parent. I will write like this: parents. So that is first one is a tall, and another one is a dwarf. Tall and the dwarf. The two parents are the monohybrid. But the two parents cross with respect to the only one contrasting character. This is one contrasting character. Meant for stem length. No, tall is also length, and dwarf is also length. It's height. So tall and dwarf, and then we have to give what alleles. Now these two alleles, capital T, and small t, small t. Now once again I have to tell you about the allele. What is allele? See, like this only you have to write. Arrows also you have to put. What are the alleles? The pair of gene. Capital T, small t, forms a allele. So means in every living being, plant, animal, human being, here the one character exhibited by what? The two gene, and that is called alleles. Understood what I am telling you now? So in every organism, including the pea plant, a single character expressed by how many genes? Two gene, and that is called what? Alleles. That is why for a tall I have written allele ಅಂದ್ರೆ ಎಷ್ಟು two genes are there. ಎರಡು genes ಇರ್ತಾವೆ, ಎರಡು genes ಇರಲೇಬೇಕು. ಒಂದು allele ಅನ್ನು ತೋರಿಸಬೇಕಂದ್ರೆ ಎರಡು genes ಇರಲೇಬೇಕು. So two gene here, capital T, small t, and then we have to separate the gamete. Separate the gametes. To this gamete. Here, what are the which are the gamete? But if it is separate, this is diploid. This both are the diploid. Diploid means the what two sets of chromosome present. So two sets of chromosome means here that's why it is shown the two gene, and here is also two gene. Here is also. So that is how to separate the gamete means diploid means capital T is a you have to always do like this. Round up capital T, and then another is small t and small t. So these are the gametes of the tall plant 2, and these are the gametes of the dark plant 2 small t small. Then you have to go for the F1 generation. F1 generation. So then F1 generation what you have to do? F1 generation you have to do like this. Capital T small, then capital T small. See what these all. So capital T small t, and here you can write all are tall. Why all are tall? Why all are tall is because here capital T dominant over the T, small t. Capital T dominant over the small t, and that is why here only one is expressed. Which one? Dominant property is only expressed, and small t is not expressed in front of the what? Capital T. And so that is why this law is called as law of dominance. I think you understood. What is the law of dominance? So law of dominance means when during the crossing of the two parent, no, and only one character expressed and another one what? Hide. Another one is what? Hidden. Means the dominant character not what being able to express the recessive character. Recessive character express ಮಾಡಲಿಕ್ಕೆ ಅವಕಾಶ ಕೊಡೋದಿಲ್ಲ. So only one character expressed, and that character generally a dominant, and that is why it is called the law of dominance. So means Mendel the concept in which the first law he derived is called what? Law of dominance. So law of dominance means when two parents are crossed, during which only one character not expressed, another not expressed. How many characters are there? Capital T means tall, small t means tall, but both were when crossed, the dwarf character not expressed, even the dwarf gene is present, and here only one character expressed. So means here what the capital T suppress the counterpart. Capital T suppress its counterpart, means it is not allowed to express the one, it is a counterpart. So that is why only one character appeared, and that means all are what? Tall. All are tall. And he is again very curious about what? He is keeping all this for the self-pollination, and waiting for the second generation. Means what? The F2 generation. F means here filial, that you can also get from the book also, and the notes is also given. And F means the first filial generation, and in the first filial generation what happened? All are tall, even the small t is present, it is not expressed. Then this same plant will be kept for self-pollination, and once again we have to separate now. Here P2 gamete, this is considered as a P1 gamete, and then next one is considered as a P2 gamete. So now P2 gamete what? So P2 gamete means now see capital T, and then small t, and then here again capital T, and then small t. P2 gamete, correct? And then what? Next is the F2 generation. Now you take the capital T, correct? And then capital T, small t. Next capital T, small t, and then small t. Correct? Now see here, this is capital T tall, this is capital T small t also tall, then this capital T small t also tall, and this is what? Dwarf. Small t dwarf. Now Here sir, 3 are tall and 1 is dwarf. So means here 3 are tall, this is 3 and this is what? 1. Correct now? So means here we have to write the ratio, and that is phenotypic ratio. Phenotypic ratio is what? 3:1. Phenotypic ratio is 3:1. Phenotypic ratio is 3:1. And then below another ratio you have to write, and that is called genotypic ratio. So now, how do you write a genotypic ratio? See, genotypic ratio, tall. This is a tall, correct now? This is a tall, but it is a homozygous. Homozygous. And this is two tall same are there, and this is called what? This is called heterozygous. Heterozygous. And this is again homozygous. Same. Homozygous. Correct? So means here what is the phenotypic ratio is how much? Phenotypic ratio is 3:1 and genotypic ratio is 1. So this is a tall homozygous is only one type. Correct? And these two tall, no, capital T small t is a similar type. So means it is a heterozygous. The 2 is there and this is again 1. So means 1. So means genotypic ratio of the monohybrid cross is 1:3:1. Now let me back to the what is a homozygous condition and the heterozygous condition. So homozygous condition means if in any plant the similar two type of genes are there, both genes are same in an allele or allelomorph, and is called as what? Homozygous. Homozygous. Understood? So means in a zygote, similar genes are present, and that is capital T, small t, same, and it is called homozygous. So that is why. So see here, this is a homozygous, and this is also homozygous. And when the two different genes present in a zygote, and that is called as heterozygous. So homozygous and the heterozygous are the two conditions. So that is why I have written here. So capital T is homozygous, small t is again homozygous, and capital T small t are the two heterozygous. So that is why genotypic ratio. So this is one type, ratio 1. These are the two type, ratio 2, and this is the one type, 1:2:1. Then why it is genotypic ratio is written 3:1? Why it is written 3:1? The 3:1 is because 3 are tall, whatever its genotypic identity, and this is only one dwarf, and that is why 3:1. Then once again I have to come back for what is the phenotype and the genotypic ratio. What is a phenotype? Phenotype means external identity of an organism. Phenotype means what? External identity of an organism. What is the look of the plant? How does it look? Either the tall or dark or colored or not colored, the black and white, or whatever what you see, and that is what? Phenotype. And by that, for example, you see, if I write here, this is a 1 and this is a 1, what you say? So you say this is a tall because it is a tall, and you say this is what? Dark, small, and it's a dark. No, so like this. And for example, phenotypic ratio says again, this is a seed. This is a round seed, and this is what? Very wrinkle and small seed. So means because of phenotype. No, then the external identity of an organism is called a phenotype. Then what is a genotype? So genotype means it is a gene identity. Means it is internal identity. So means internal identity, what is there, on that base the phenotype. Got it? So means what type of genes are present in any organism. What type of gene present in an organism, and on that base the phenotype. So that is why. So phenotype depend on what? The genotype. So genotype means the internal character, and phenotype means it is a external character. So this is about the one gene inheritance. So one gene inheritance or the monohybrid cross. So means I think you understood crossing of two parent with respect to the only one contrasting character. So the ratio of the phenotypic ratio is 3:1 and the genotypic ratio is 1:1. So if you complete this in the examination, it is asked, and for that 5 marks is there. See, everywhere marks you have to write it correctly. Particularly you have to write gamete always in what this whole bracket means the round bond level. So like this you have to write. And book also another type is given. Another type of this in the book they have given what for crossing of this pair. For example, I will show that they have given like this. See, if it is a capital T, small t, capital T, small t. If it is crossed, cross and they have made the checkerboard system like this. They have made the checkerboard system, and one gamete writing here, capital T, and then another small t, and then here capital T, and then here small t. Gamete are separated, capital T, capital T, capital T. So capital T, small t, then capital T, small t, and then last one is small t, small t. Like this in book it is given, but what I have shown is the easiest method instead of making checkerboard. And in monohybrid you will not require to use the checkerboard, but in the dihybrid definitely you have to use the checkerboard system. Next one is test cross. Then what is a test cross? So test cross means it is to know the homozygosity or the heterozygosity of the F1 plant or unknown dominant plant. F1 plant or unknown dominant plant. So in unknown dominant plant, why we have to know? Because see, if I say, and there is a two condition may be there. One is capital T is also tall, and capital T small t is also tall. No, so two conditions are there tall. Then we cannot understand the taken plant is either what a homozygous or heterozygous. Taken plant is either a homozygous or heterozygous. So means capital T, small t. If the two conditions are there for the tall, so this is also tall, this is also tall. But when I write, I know it is a homozygous and it is also. But see, if I give the diagram of the tall, this is also tall, this is also tall. Can you tell now what is the genotype of this condition? No. Genotype of the condition, you cannot say no. So in such a condition, what you have to do? See, for example, while crossing capital T, for example, capital T, if I cross with a small t, and see capital T, small t means you will get all are tall. This is one condition. If another condition is there, capital T, small t, this is one plant and this is another plant I am giving. So capital T, small t, so this is what heterozygous, and small t. If I cross capital T, small t, see small t, so means the result different. No, all are not tall. So means 50% are tall and another 50% dwarf. So means here ratio is 1:1. So when the ratio is 1:1 means taken the unknown dominant plant is heterozygous. I think you understood. The unknown dominant plant taken is heterozygous and ratio is all tall means the ratio is 1 means here the unknown dominant plant taken is what? Homozygous. I think you understood. So when homozygous means all must be one type plant. Heterozygous means the 50% one type and 50% another type. So means two different gametes are present here. So that is why heterozygous condition. So we'll stop here only, and tomorrow we'll go to